Hi guys, I'm Changyuan Wang. I don't have other team members, so I will deliver this presentation by myself. I am more of a soft material person, so my topic is closely related to biophysics. Today, I am going to talk about microtubules and their mechanical properties. I will abbreviate microtubules as MT in the following slides. So first, what are microtubules? Microtubules are one of the components of the cytoskeleton. We know that our skeletons are hard. Some of the bones are even harder than concrete. Microtubules, which act like the skeleton of the cell, are also really tough and stable comparing to other parts of the cell. Microtubules and other filaments form a network to protect the cell from deforming too much. Their diameters is typically about 10 to 13 nanometers, but they can grow to about a few microns or even 50 microns. So if you look at their global pictures, they really look like thin strings and noodles, but tough noodles that cannot be easily broken or ruptured. They are highly dynamic. If you look at small movies of them, these strings will swim rapidly like worms all the way around. These days, people are paying more and more attention to microtubules. They study it in researches, study single microtubule. They look at it at bundles of microtubule and even a bulk of microtubules. After the physical properties have been quantitatively measured, microtubules start to be applied in the field of bioengineering, and we will introduce some applications later on. Today, we will mainly focus on the stiffness and the stability of the microtubules. We will talk about both the lateral deformation and the elongation of them. Like I just said, microtubules are the parts of the framework of the cell, so we already know that it is tough. So how can we quantify how stable it is? We need to use some technology to measure them. We will see some methods that we have talked about in class, like indentation and AFM. We will discuss how researchers study microtubules, what type of methods they use, what are the significance of these properties, and their corresponding applications. So first, let us have a brief understanding of the microtubules. In the right figure, we can see that we can decompose a microtubule into little strings, and the string is connected by single particles. The strings are called protofilaments, and 13 protofilaments together form a tube, which is the microtubule. However, the microtubule is not an isotropic cylinder. The particles inside the protofilaments, which are the particle in the z direction, have stronger interaction than those particles in the xy plane, which are the adjacent particles in different protofilaments. So when a microtubule decomposes, they usually first unzip into different protofilaments and then become single particles. After we know the structure of the microtubule, we can use a hand-wavy way to understand its Young's modulus and shear modulus. Young's modulus is the resistance of protofilaments. It's like if you stretch the filaments, to what extent you can resist the tension. Shear modulus is closely related to the weak interaction between protofilaments. Some recent researchers have been discussing that maybe if you don't match the protofilaments one by one exactly, so it's like if you mismatch one or two particles and shift the protofilaments by 2 or 3 units, just like the figure below, they may still form a stable configuration and just stay in that structure. We just said that microtubules are more vulnerable in the lateral direction, so researchers are usually more interested in lateral deformation than the elongation of the microtubules. The reason is the protofilaments themselves are actually pretty strong because microtubules usually fall apart in lateral directions way before the protofilaments decompose into single particles. So if you apply the microtubules in bioengineering, you need more quantitative data about lateral deformation. You need to make sure that how much pressure they can take in the lateral direction. So usually, if you apply the pressure isotropically, you won't need to worry about the elongation because like the protofilaments are pretty strong. This is one of the reasons why there are less paper about the elasticity of the microtubules, but much more paper about their lateral deformation. Okay, now we know the lateral deformation is important, so we need to introduce another parameter, bending modulus. We want to suspend part of the microtubules, use an AFM tip to deform it, and the bending modulus is the parameter to describe the extent of deformation. The researchers have found that the bending modulus is closely related to Young's modulus, shear modulus, G, and some geometric factors. Fs is a constant, it is the shape factor. D, which is the diameter, the external diameter is about 25 nanometers, and the internal one is about like 15 nanometers. And the bending modulus is also length dependent. It depends on the suspended length L. Since almost nobody cares about the elasticity of the microtubule, so it's actually pretty difficult to find a constituent law for microtubules. More quantitative measures are about the lateral deformation, so which is like the bending modulus formula on the slide. Okay, the bending modulus is actually temperature dependent. If we look at the right figure, the orange line is the bending modulus curve in 37 degrees Celsius, and the blue line is in 7 degrees Celsius. The microtubule appear to be more compliant at higher temperature. They're easier to deform. The main reason is the shear modulus, G, is 12 megapascals at 37 degrees, but only 1.9 megapascals at 7 degrees. From the number, we can see that there is a strong temperature dependence of the lateral interactions between protofilaments. This actually makes sense because it implies that the weak interactions between particles depends on temperature. Higher temperature means higher energy, so it's relatively easier to alter the bonds. This is not counterintuitive. It actually makes sense. There are also other methods to investigate the lateral deformation. One of them is indentation, which is a method that we talk about in class. For indentations up to 3.6 nanometers, which is about 15% of the microtubule diameter, there is approximately a linear elastic response. If we unload it, we can see the process is reversible and there is no hysteresis loop. If we continue to increase the force up to about 0.3 nanonewtons to 0.35 nanonewtons, microtubule will experience a small collapse and there will be a 1 nanometer indentation step. This time, the process is no longer reversible and a hysteresis loop is created. If the force goes even higher, it may lead to substantial damage to microtubules. For example, the depolymerization. The microtubules may fall apart. They may become such long and thin protofilaments or even fall into small single particles. 0.3 nanonewtons may seem to be a small force for like metals and crystals, but it is actually a pretty high value for such a small piece of biomaterial. Comparing to other soft matters, microtubules actually takes on like a pretty impressive value of deformation. Is relatively strong. Okay, but what about the 1 nanometer indentation gap? Why there is such a gap? Researchers have made some speculations. One hypothesis is the small rupture of the tubulin bonds. The tubulin protein is the fundamental building block of microtubules. If you look at the picture in the corner, tubulin is actually a single small protein molecule that made up the whole microtubule tube. The irreversible process may due to the rupture of the bonds between tubulin, maybe the axial bonds, the rupture inside the protofilaments, or maybe the lateral bonds break, like the adjacent tubulin protein fall apart. Lateral bonds have a higher chance to break because they are 5.2 times weaker than the axial bonds. So more likely the microtubules will unzip, like the picture in the upper right corner. After the microtubules unzip, they may reannihilate. The protofilaments may shift along the axis for about 4 or 5 units. Since the tubulin dimers, those proteins pairs are identical. So when we shift them, they can have weak interactions between each other and form an alternative stable conformation. Yeah, because like it's identical. If you shift them, they're still like the same thing. This is one of the reasons why we can see such jagged up and down tracks in the force indentation curve. The reason is like the bonds between proteins first break and then reannihilate and form a bond again. Okay, we just talked about some reasons why there is a 1 nanometer gap, and now we can directly look at the gap. Like what we talked about in class, we can do an integral, and the shaded area is the work needed to load and unload the microtubule. The hysteresis loop is approximately the triangle with an area about 1.35 times 10 to the power of negative 19 joules, which is about 32.8 kBT. The energy dissipated during the load-unload process is not too large, but we can still clearly see the difference between the load and unload curve. One thing I found interesting about soft material is the ability of self-healing. If we limited the force to about 0.3 to 0.4 nanonewtons, we can investigate the behaviors near collapse. For the force about 0.27 nanonewtons to 0.3 nanonewtons, we can see that the AFM tip deformed the microtubule a little bit, but after we recheck the force, the microtubule recovers. This is a reversible process. It's always reversible. Sometimes, even if the microtubule already collapsed, they may still slowly reannihilate. The holes on the microtubule may be recovered by themselves if there is no other free tubulin in the solution. Self-healing is actually pretty interesting mechanisms for soft matters like polymers and especially some biopolymers. Another interesting thing in technology is AFM imaging. Since microtubules are small, soft, and vulnerable, by vulnerable I mean compared to metals and crystals, they are we need to take good care of them. We need to make sure to minimize the damage induced by scanning. So during the experiment, we operate the AFM in the jumping mode. This is the most delicate AFM mode. Basically, it is like the lateral movement of the scanning tip with respect to the sample surface is performed when two of them are not in contact. In other words, if you look at the figure, the top one is actually is what we usually do. We drag along the sample and do the scanning. Jumping mode is the bottom figure, which is like we lower down the tip, do the indentation, lift up the tip so that it stays above the surface, and then move forward to another point. In this manner, lateral shear forces on the specimen are minimized. In our experiment of microtubules, the tip is elevated to about 30 nanometers from the surface before we move forward to the next point. We know that the diameter of microtubules are typically like 25 nanometers, so usually is smaller than 30 nanometers. So we indeed are minimizing our lateral drag forces on the sample. The jumping mode of AFM can also do a stiffness map. To visualize the stiffness distribution, we adopt a development version of the WXM scanning software. For each scan point, we perform a force distance curve with a force limited to about like 100 piconewtons to prevent the damage. After we do a linear fit, and the software will help us calculate the stiffness and show the data in the stiffness map. We can see in the right figure, the darker color represents the softer regions. We can see that the microtubules are clearly softer than the background, and the stiffness is almost homogeneous over the surface, except on the boundaries. The boundary is darker, meaning that the sides are softer, and away from the boundary, the stiffness is pretty uniform. And yeah, it makes sense. Okay, we have talked about enough experiments in the lateral directions. Now we can briefly mention some interesting phenomena in the axial directions. We will do it in a qualitative way. If we decompose microtubules, we can see that the fundamental building blocks are the tubulin proteins. We just talked about it. There are actually two kinds of proteins, alpha type and beta type. The fact that microtubules are asymmetrical in the molecular level make it exhibit the property of polarity. One end has only beta subunit exposed. It is called the plus end. It has a positive charge. And one end has only alpha protein exposed, and it is the minus end. When we stretch the two ends will elongate at different rates. So how different they are. Let's look at the picture from the video light microscopy. It is taken under DIC. DIC in this context is not digital image correlation. It stands for differential interference contrast microscopy, which is a pretty common microscopy method in biology and biophysics lab. We typically use it to look at some biological samples, and some condensed matter lab will use it to deal with some semiconductors like silicons. Usually, it won't be used to test the elasticity of microtubules. So this method may appear to be a little weird for some mechanical engineers and some material scientists, but we're not doing it in a quantitative way. We're just looking at it intuitively. So in the figure, there is a single microtubule, and we can try to elongate the two ends at the same time. If we look at the right end, we can see that it slowly grows longer and longer, and it continues to stretch for 84 seconds, and suddenly it shortens. In contrast, the left end doesn't seem to move like the left end. Yeah, there's maybe a little change, but it's not obvious. The visualization tell us that the plus end elongates much more rapidly than the slow end. Yeah, it's like pretty obvious if you look at the pictures. So what is the significance of it? Why we care about the difference in elongation at two ends? One of the reasons is it explains some behaviors of microtubules in the cell. Since microtubules are structurally asymmetrical, the molecular motors on the microtubule will move in one direction. For example, kinesin and dynein are two types of biological motor proteins. They actually have two ends that look like feet. They will walk on the microtubule. Because of the polarity, they will only walk unidirectionally. Kinesin will walk towards the plus end, and dynein will walk towards the minus end. In this way, the asymmetrical subunits help explain the behaviors of the motor proteins. It also has some engineering implications. We can attach cargoes on the motor proteins and let them walk on the microtubules to do some transportation. We can also fix the motors and let the microtubules walk. Yeah, do it in the reverse way. We have just mentioned that some experiments in the lateral directions and the lateral stiffness set up a limit for the weight and the amount of cargoes that we can attach on the motors. This is an example of bioengineering in nanoscale transportation. If we use materials to make those small nanoscale devices, it will require more work and more money. But microtubules are cheap and they can be easily attained. So and they're soft and stable, so they're like good bioengineering materials. Microtubules is also an ideal 3D isotropic active fluids. For example, in our lab, we study active matter and self-assembly. We are interested in how microtubules. We are interested in how biomolecules, biopolymers in general, assemble by themselves under certain conditions. For microtubules specifically, if you add certain concentrations of PEG and salt, the microtubules will come together and form bundles. Those bundles of microtubules will form a pretty stable, long-lived, extensive active fluids. We can see that the fact that microtubules are pretty stiff and stable make it a nice material for bioengineering. And since bioengineering needed, it intrigues more scientific researchers on the properties of the microtubules. And yes, these are all I wanted to talk about. This page includes all the paper that I cited, and thank you for your listening.